working on it even before this project, before this Improving Pig Livability project. And it really started in 2017-18 when we started developing a large national survey to collect information from a little over 104 sow farms in 13 states across the U.S. They were large farms, integrated farms, independent farms, small farms, and really we wanted to have a good assessment across systems, across databases that are utilized. What is mortality looking like, and how can we wrap our heads around, or start wrapping our heads around pelvic organ prolapse? And so these are really the farms. We really collected data for every week for a year on about 385,000 sows worth of production. And here you can see a bar chart for each of those farms, right? These are anonymous or randomized IDs. But what you can see is that some farms were running over 20% on sow mortality. Some farms were running below 5% for the entire year, which is exceptional, right? And what we really see is that this 12.7% was the average for that year on total mortality. And pelvic organ prolapse-induced mortality was about 2.7% of that, or actually around 21% of all mortality in the sow farms across the U.S. was occurring due to pelvic organ prolapse. So when you break that down, what contributes to that 21%? Well, 15% of those are vaginal or uterine prolapse, 4% of those were rectal or anal prolapse, and 2% of those were both of those happening, that were resulting in either the sow being found dead or having to be destroyed. So what were the outcomes of that? We collected all kinds of measures from those sow farms, really to try to give us an indication of where we wanted to create and prioritize hypothesis-driven research. And some things that we found that weren't really related to pelvic organ prolapse in this data set were, tail length of the sows from docking as a piglet, induction protocols, particle size in the feed, herd size. Those were things that we looked at and did not see a relationship. There was a lot of things that we thought about, genetics, antibiotic usage, health status, mycotoxins, geographical region. These are all things that, may have an influence, right? There's a little smoke there, but we just didn't have the data set and the horsepower and the information we were able to collect to be able to get a solid answer on that. And then there were things like managing the sow's body condition, the bump feeding strategy, and really developing a perineal scoring system that we think were really strong outcomes of that research, and that helped guide some of the work that we continue to build on today. And so a little about that perineal scoring evaluation system. What we did when we were visiting those sow farms is we scored sows in late gestation and assigned them one of three scores, a 1, 2, or 3. And a 1 really indicated that those sows we thought were at minimal risk. We considered that to be phenotypically normal, where there's a normal swelling and protrusion, right, that comes with late gestation. But a 3 would be these sows that have significant protrusion in the perineal area, both in the rectal area as well as the vulva area. And so as we scored those sows, what we found out is that sow farms with a higher average perineal score also had a higher prolapse incidence rate, right, compared to those that were lower. And so individually, those sows that had a 3 were much more likely to prolapse than the sows that scored a 1 or a 2 also. And so that really told us that there's some biological underpinnings that are happening in the sows that have a high perineal score that are either the result of the high score, right, or causative of the high score. And so we're still not quite sorting it out yet from a causation standpoint. But that really was the driver in our objective in this large grant, to determine the physiological, endocrinological, nutritional, genetics, microbial. What are those factors and how are they associated with pelvic organ prolapse and the development, the phenotypic development of becoming high risk? So one of the first places we started to look was really trying to better understand the vaginal microbiome of sows that were low and high risk. And the reason we were interested in this was because in other species, in humans as well as cattle, there are issues, right? There are diseases, pelvic inflammatory disease and bacterial vaginosis that in humans also causes a weakening of the broad ligament and can lead to prolapse. But that's not been well characterized in pigs. So Zoe, my graduate student at the time, and still a graduate student, has started working on that to better understand those sows that have low and high risk, do they differ with respect to their microflora in the reproductive tract. And really what she found in this first study, so I'm going to go through about 6 different studies that we've looked at in the course of this project and just try to give the highlights, because we only had 15 minutes for this part, try to give the highlights of what we've been looking at, what we're learning, and try to indicate a little bit about where we're going. So on this first study, what we saw is that we had a group of sows that were perineal score 3 and a group of sows that were perineal score 1, of which 1% prolapsed and 23% of the PS3 or high-risk sows prolapsed. And we sequenced their vaginal microbiome. And what we found is that there are actually specific bugs, and these OTUs that you'll hear me, this stands for Operational Taxonomic Unit. And that really refers to a different genus or species of a bug. And what we found is that when comparing the high risk to the low risk sows, that there were some that were increased in the low risk sows or higher, like Veillonella, and there are others like Streptococcus dysgalactiae and members of the Treponema and Prevotella families that were greater in the perineal score 3 sows. So those were that was our first observation, right? That there was some support of that hypothesis that the vaginal microflora could be different between sows that are at different levels of risk. And so we wanted to follow that up with a second study. And so then a year and a half later, we went to a different state and used 2 different sow farms and different genetic backgrounds to determine if we could validate some of our initial observations. And so this schematic here kind of shows the experimental design, but really what we did over the course of 7 weeks was collect vaginal swabs from sows for 4 weeks worth of gestation. So all sows that were week 12 through 15 were collected, and we did a perineal scoring on week 15. And actually, we did a perineal scoring every week. And what we were able to see is that perineal score, where they become high risk, really manifests in that last 2 to 3 weeks of gestation. And at week 15, 4.5% of those sows would be considered high risk, of which, in this study, 23% of those subsequently experienced a pelvic organ prolapse. So on this, when we go back and analyze, sequence the microflora using 16S ribosomal DNA sequencing, identify their genus or species that they represent, we saw, again, 18 bugs or OTUs that were more abundant in the low-risk animals and 33 that were more abundant in the high-risk. And you also see some repeat offenders, right? Streptococcus dysgalactiae, Treponema. So those were interesting, the validation that some of the same ones in a different study were also statistically different in the same manner that they were in the first study. So we also looked at those that were high risk that prolapsed or did not, and we also saw that of those that are high risk, Veillonella, which again is typically greater in those sows that are low risk, was greater in sows that did not prolapse compared to those that did. So that may be a potential for a feature or something we can look at from a biomarker standpoint to recognize reproductive health. The third study that we've been working on used the same animals, except we drew blood samples from the same ones that we collected vaginal swabs on. We did a CBC analysis. We looked at different biomarkers associated with inflammation, and we looked at steroid hormones in those animals. And one of the things that we saw initially was that there was a significant decrease in the lymphocytes and the monocytes of those sows that were at higher risk of pelvic organ prolapse, right? And if you go back to that picture I showed you, without validating it, our working hypothesis is that some of those tissues, if there is an infection or an issue with those tissues that cause lymphocyte migration and trafficking to those tissues, you would have a reduction in those that are in circulation, and there's some literature to suggest that that happens in other events as well, or other health challenges. We also looked at LPS binding protein. That was the only one of the biomarkers that we really looked at and studied. That was increased in the perineal score 3 sows, being about 25% greater in those that were high risk. We saw an increase in steroid hormones, so estrogen and androstenedione and aldosterone were all increased in sows that were high risk. And so when you click through, some of those first 3 studies, we really saw there was changes in the microflora, and there was changes in markers of inflammation, and that was really what we were associating with high-risk sows. And so we followed that up with a couple different studies that we really wanted to see, okay, well, is there strategies that we can use to shift or alter the microbiome and also alter the outcome of pelvic organ prolapse? Well, we also wanted to, before we did that, actually, I'm going to show you 2 slides before I show you the couple interventions that we attempted. And one of those was also looking at the fecal microbiome. And what we did there was the same approach that we did on the vaginal microbiome, but we wanted to determine if it was different or similar. And what these plots over here, these really what they show you is that the vaginal communities are quite different between the fecal and samples and the vaginal samples. And if you look at the overlap of species, what we see is that there was, out of the roughly 10,000 organisms represented, 234 of those were unique to the vaginal microbiome community, and 1,100 were unique to the fecal community. But what we did see that was interesting is that Clostridiums, Treponema, Streptococcus, Veillonella, some of the same things that we saw were different based on risk of prolapse in the vaginal microflora were also different in the fecal microflora. So the fifth study that we worked on was using on-farm mixing of BMD, and this is something that was, others had used it and had anecdotally thought that there was some value in using this and mixing it in the feed on farm. And so we designed a study to do that at 2 different sow farms, one using the water soluble and one using mixing it in the feed on the farm. And we used roughly 2,000 sows at 2 different farms. And at the end of the day, what we saw was that there was no difference on prolapse incidence. And we used that intervention during the last 2 weeks in gestation. And so while we didn't see a difference on pelvic organ prolapse, we did see a difference in the number of stillbirths that were happening. And so one thing that we've seen consistently over projects is those sows that are high risk for prolapse also have significantly greater stillbirths. And so that's one of the things that even if you're thinking about from a management standpoint, sows that are at risk for losing pigs due to stillbirth, it would be those sows that are also at risk of having a pelvic organ prolapse. So not all of them prolapse, right? It's still a relatively smaller percentage. Anywhere between 10 and 23% of those will prolapse, but they're losing more than a pig extra due to stillbirth. So we also try to study where we use vaginal infusion of ampicillin during late gestation. So rather than feeding a product, we went through and 3 weeks and about 3 days, we did a double infusion. And roughly, again, around 1,000 sows, some controls and some infused twice. And the idea was really actually closer to 1,500, 739 controls and 824 that were in the treatment group. And at the end of the day, if you look at the perineal scores, we did not shift the perineal scores in those farms at all based on the infusion of ampicillin. And we also didn't shift the frequency of pelvic organ prolapse. And this kind of shows the pelvic organ prolapse rate by perineal score 3. So as the score goes up, we saw an increase in incidence rate, but we did not see any effect of the infusions, unfortunately. So again, in that study as well, this is different sow farm. Again, on this study, different genetic backgrounds. Those sows that are at high risk had substantially more stillborn piglets, almost a pig more than a pig compared to the low-risk sows for prolapse. So those are some of the 6 studies that we've been working on. There's a 7th one here that we're in progress right now. Dr. Jack Deckers has kind of joined our group. He's a molecular geneticist at Iowa State University, and we're collaborating with Topics Norsvin, who's provided a tremendous data set to Jack's PhD student, Vishesh Bhatia, who also has a poster here, by the way, that you can get into more details on this, and Vishesh can give you a lot of updates on what we're doing. But really, that data set is records from almost 38,000 sows over a 10-year period of time that had an experience of about 7% pelvic organ prolapse. And this is just the summary slide here, but really what Vishesh has shown so far is there's about a 17% heritability associated within these lines associated with risk for pelvic or actual pelvic organ prolapse. So he's working through that now to refine those analyses and continue going through and looking for specific genomic regions that may be associated with that outcome. So again, quick summary, the vaginal and fecal microbiomes differ based on the risk of pelvic organ prolapse in sows. On-farm mixing of BMD did not influence incidence of POP incidence, although it did have a relationship or had a capacity to reduce stillbirths in the experiment where we used it, particularly in the sows that were high risk for prolapse. Vaginal infusions with ampicillin did not affect pelvic organ prolapse. It also did not affect the perineal score distribution in those farms. And there does appear to be a heritability associated with some lines for pelvic organ prolapse, and finding out how this manifests and understanding the relationship with the environment of those pigs. To be able to express the phenotype of having a prolapse, there has to be an environmental interaction, and we're still working to identify what some of those pieces are that may help to create that understanding, the biological underpinnings that drive us out to die due to pelvic organ prolapse. So lots of collaborators on this project. I had topics on the last slide, but you can see we've worked with a lot of people, a lot of farms. If you've worked with us, and I didn't have your sign up here, I apologize, but throughout this last 5 years, there's been a lot of people who have really contributed in a big way, and I really think it's partly because the industry is so uniform in working towards solutions in pig livability. So thank you very much, and I think we'll do questions maybe as the panel at the end.